Hello everyone and namaskar to one and all. This is chapter 10 part 5, last part of this chapter, and in this part we have to discuss some typical numerical problems which has come many more times, and you know it is the last part of chapter. We have to see SCRT exemplars, and then in the last we have to see previous year examinations questions, some of the important questions. This is chapter 10 part 5, last part of this. Remaining part of the chapter we'll discuss in this, and we'll discuss many important numerical problems which are frequently coming in competitive examinations. How to solve these problems, the tricks and tips I'll give to you. And as this is the last part of the chapter, as usual we will discuss some NCERT exemplar questions, and then we will say that is previous year examinations question. This is part that is 5 of chapter 10. You see here, in case of plants especially, that is meiosis. In meiosis, cytokinesis that can be of two types. One is called successive type, another is called simultaneous type. You know children, basically in meiosis what is there? That is, a cell will divide and ultimately initially the two cells will be formed, and then there will be, that is, four cells will be formed. You know, as and when that is karyokinesis is over, there will be cytokinesis, and there are two divisions, first and second. So it is assumed there will be two times cytokinesis. One cytokinesis will be after meiosis first, and one cytokinesis will be after, that is, karyokinesis of meiosis second division. But see, some variations are there. Successive type means what? That is, in this type cytokinesis occurs after each nuclear division. You can see here, first you see that this is the cell, this is nucleus. That is, the nucleus has gone under, that is, two cells have been formed. That is, this was having 2n number of chromosome. This is meiosis first division. Two nuclei have been formed. Each is having n and n number of chromosome, and there is cytokinesis. So meiosis first is followed by cytokinesis. Two haploid cells have been formed. Now again that will divide. The same thing will happen here. That is, it will divide into two nuclei like this and this. It will also divide into two nuclei, and after this second meiotic division, they will also divide into two cells. Therefore, the four cells will be formed. So each, how to differentiate? That is, cytokinesis is after every nuclear division, first nuclear division and second nuclear division. The four cells are formed by successive cytokinesis. These are either arranged isobilaterally like this is isobilateral arrangement, or they can be arranged in a linear fashion like this, linearly arranged. You can see the diagram here. This is first meiosis, two cells have been formed. Second meiosis, four cells have been formed. So this is isobilateral arrangement, or they may be arranged like this, that is, these are linearly arranged. This type of arrangement usually is found in case of, that is, monocote angiosperms, and it is said that this is advanced type of, that is, cytokinesis, or advanced type of, that is, meiosis division. Is it clear? What is simultaneous type then? Cytokinesis takes place at the end of both division. What is there? That is, this is first division. Two nuclei have been formed. This is second division. Four nuclei have been formed. That is, one this side, one this side, one this side, one will be on back of this. And when four nuclei have been formed, that will be followed by, that is, cell plate formation or cytokinesis. So these four will be arranged like this. That is, these are arranged in a tetrahedral way. These are arranged like this, 1, 2, 3, and the fourth will be that side. So this is called simultaneous type. It results, that is, the arrangement, four haploid cells are arranged in tetrahedron. Such type of arrangement is usually seen, most common this is in case of dicote, and this is comparatively is treated as, this is called primitive type of, that is, cell division or cytokinesis. Is it right? So you have to remember, in case of plant, cytokinesis, successive type, and that is cytokinesis may be simultaneous type. This is usually observed at the time of, that is, microsporogenesis. That is sporogenesis. You know, microsporogenesis is formation of, that is, microspore. Microspore means in time to come they will develop into pollen grains. You know, pollen grain is the first cell of, that is, male gametophyte. When the pollen grains are to be formed, or what is there, that is, microspores are to be formed, that is, this division will take place. Meiosis will be there. As a result of this, the 4 haploid cell will be formed, that is either by successive type of cytokinesis or by simultaneous type of cytokinesis. Is it okay? Now children, we have to see some very interesting, that is, numerical problems based on theoretical, that is, background of meiosis and mitosis, and that is interface. You have to see one by one. You have to be little, that is, extra conscious here, because this is very much logical one. You should not get confused anyway. See the first question. What's the number of chromosome and DNA amount in pollen mother cell of G2 phase if the pollen grain has 10 chromosome and 5 picogram DNA? Start from here. What is given to you? Pollen grain. You see children, these are the 4 pollen grain, 1, 2, 3 and 4. These are haploid number of chromosome they have. What's the haploid number of chromosome? Each is having 10 chromosome, 10 in this, 10 in this, 10 in this, and 10 in 10 chromosomes we have. Amount of DNA in each we have, that is 5. This is DNA content, that is 5. The unit is picogram. So he is asking pollen mother cell. Children, you see, that will be the pollen mother cell. First it will divide, that is, into 2 daughter cells, and then it will divide like this into 2 and 2. Right? You see here 10 chromosome. Let we are taking, that is fate of 1 chromosome. This is 1 chromosome represented by what? 1 chromatid. Is it right? So this is having 10. It is having 10. So how much that will be here? That will also have number of chromosome will be 10. Chromosome will be 10 chromosome here. But what's the difference? Here the chromosome is represented by 1 chromatid. Here the chromosome will be represented by 2 chromatid. Is it? But here the chromosome is having 2 chromatid, having 1 chromatid. So in this mother cell of these two, number of chromosome will remain same, 10. But here amount of DNA 5 in this, 5 in this. The total amount of DNA will be here. That will be 10. DNA will be here. Is it right? 10C. Clear? Come to the, what will be here now? This is pollen mother cell. That is 10 chromosome of this has gone to this side, 10 has gone to this side, because there is reduction division. So what will the number of chromosome here? Number of chromosome, that will be 20. And you see that the chromosome is represented by, that is 2 chromatid. What will be amount of DNA? Amount of DNA, because half chromosome have gone to this side, half have gone to this side. Therefore, here amount of DNA 10 plus 10 cDNA this side, that will be what? 20 amount, that is 20C will be the amount of DNA. Is it right? No problem. But you see what he is asking is not the question. You have seen in pollen mother cell, which is just to divide, it will have 20 chromosome and DNA amount will be 20C. But he is asking, that is pollen mother cell at G2 phase. You see here G2 phase, we can see here that this cell is ready to divide, and it has just completed here, that is this cell cycle. So here you see in G1 this is S and this is G2. Can you find out at G1 S and that is G2, number of the chromosome will remain same, is it? So what will the number of chromosome as G2? Number of chromosome in that will be 20, and amount of DNA will also be here, that will be 20. And because it is just, it has to enter in this. So this at G2 phase of this pollen mother cell at G2 phase will also have 20 chromosomes, and that is 20C of DNA. Is it right? Any doubt? Yes. Come to this question. How many bivalent are formed? You know bivalent. If the number of chromosomes are 4, the bivalent will be how many? That will be one bivalent. That will be other bivalent. The number of that is bivalent will be half of, what is this? Number of chromosome in human, I told you. If this is 46, then the number of bivalent will be half of this 23. Number of bivalent formed in pygium sativum. In pygium sativum, number of the chromosome is 14. So that will be divided by 2. Bivalents will be, that is 7. Is it right? Very interesting question. The third one you see now. The DNA content of a cell at S phase, S, prophase first, prophase second, respectively of meiocyte. Very interesting question this is. Come on. That we can see it like this. You see this question here. Here. This is the cell cycle of this. DNA content cell at S phase. See, this is G1. Is it? This is S and this is G2. Now the cell will divide here. It will enter into prophase first. This is prophase first, right? Prophase second, we have to find out. Now it has divided into, that is 2 cell here. Now it will not enter into after that is meiosis second, it will not enter into interphase. But you see the phase of this. Come on. That is, if S is having 4C of, that is DNA content. In the Z2 also, that is amount of DNA will be 4C. And in prophase first, that will also have how much content of DNA? That is 4C. One question is over. If the S is having 4C, then that is prophase first will also have how much? 4C. So this is having, how much it is having? That is 4C. Now these 2 daughter cells have been formed. 2 daughter cells. That is in daughter cells, how much amount of DNA will be? That will be 2C and 2C. Now it will enter into the prophase second. Prophase second, what will be there? You see, it's not end of prophase, it is not anaphase. In prophase second, the amount of DNA will remain 2C only. No doubt in anaphase second, amount of DNA will be halved, 1C and 1C. But he is asking prophase second. The right answer will be this one. Okay. What will be the DNA content in cell at G1, G2 anaphase? Content after mitotic phase is 40. See here, G1, G2 anaphase. Content after mitotic phase is 40. Is it clear? Come on, mitotic phase is completed. See again here, this is cell cycle. This one is, that is M phase. In G1, you know, amount of DNA, in S amount of DNA will be just, that is double of this, and that will be here. That is G1, G2. Can you compare G1 and G2? Amount of DNA will be double in G2 as compared to, that is G1. You see this question here. Here G1 is 40. If this is having 40C of DNA, then in S that will be 80C of DNA, duplication will be there, and in G2 also there will be 80C of DNA content. Is it right? This part up to this is over. G1 is 40, G2 is. What will be there in anaphase? Important one. Now this cell that is with 40C DNA, it has entered into the anaphase. I'm going to enlarge here. This is anaphase. What will be there in anaphase? That is chromatids. They have gone to this side. That is like this. Chromosomes segregated into chromatid. But remember that the cell have not been formed. We are asking about this cell. So this cell, no doubt, that is out of this, that is 80. 40 has gone to this side. When it will be? Yes, that will be only when there will be cytokinesis. But by now in anaphase, amount of DNA will remain as it is. That will be 40 here. This is 80. Is it? This is G1 is 40, 80, 80. Here how much that will be? This will have, that is 80, that is C of DNA. And here also that will have, anaphase also that will have 80, that is C DNA. Because
cytokinesis has not been taken place. That is, the two nuclei have not been formed so far. The two cells have not been formed so far. Is it right? See that few more questions. Yes. Initial amount of DNA is denoted by 2C, amount of chromosome after S phase. It's very simple question, my dear children. You know, number of chromosome I can repeat it many times, and again I'll give a very clear-cut view of this question. You see here, this is cell cycle, this one is M, right? This is the cell which is dividing. The cell is having this chromosome. The chromosome is represented by two chromatids. Let the chromosome here we have, that is one chromosome, or better to call two chromosome. One chromatid is this, one chromatid. How many chromosome we have? That is two chromatid, right? The cell is having how many chromosome? Chromosomes we have two, and each is with two. Now each is with two chromatid. Now it has divided, two cells have been formed. Number of the chromosome in this will also be 2, here also be 2. But here there will be chromosome is having, that is single chromatid, one chromatid here, one chromatid here. Two chromosomes are there, but the chromosomes are with, come on, with one chromatid. What will be in G1? G1 will also have two chromosome, but chromosomes are with one chromatid. In S, again number of chromosome will be 2 here, but each chromosome will have, that is, two chromatids now. Is it right? Come to the question now. Initial amount of DNA is denoted by C. He is not asking amount of DNA. He is concerned only amount of chromosome after S phase. Amount of what? Chromosome after S phase. That is, little mistake is there. Amount of DNA. That should be DNA. Amount of, that is DNA after S phase. Can you tell me? Here is how much we have? 2C. 2C is here. After S phase, that amount of DNA will be here. Number of chromosome is same. That amount of DNA, that will be doubled of. G1 is having how much? S and G2. Can you summarize that diagram? If the G1 is having 2C, S will have 4C, and G2 will also have how much? That is 4C of DNA. Is it clear? Answer will be 4C. Number of the chromosome 46 at G1. Number of the chromosome, this question you can correlate here. Number of the chromosome, that is, will remain same in cell cycle. The only thing is, that is, in G1, yes, chromosome will have one chromatid. In S, the chromosome will have two chromatid. In G2 also the chromosome will have, that is, two chromatid. Number of the chromatid will remain same, that is 46. Number of bivalent, you can recall what I told you right now. Number of bivalent is equal to number of total chromosome divided by 2. In meiosis in human is having how many chromosome? That is 46 divided by 2. That will be, that is 23. You know, 46 chromosome. Let these are the 46 chromosome. They will arrange in pair. Bivalent will become, that is, half of this. This is equal to haploid number of chromosome. Haploid number of chromosome in human, that will be 2n divided by 2, that will be 23. So bivalent is equal to, that is, number of haploid chromosome. Right? See this last question, very interesting one. What is this? Mitotically dividing vegetative cell. You got the mitosis division is a flowering plant, makes no difference. At G1 has 20 chromosome. Simple thing, you draw this diagram. This is G1. How many? 20 chromosome. Can I make one? This is one chromosome with one chromatid. This is 20 chromosome. In S, this is also, that is 20 chromosome. That will be 20 chromosome. Here also chromosome number will be 20, but that will be with two chromatid. And here also at G2 also, can you tell me? Number of the chromosome will remain same, and the chromosome will have two chromatid. You got it now? G1 has 20 chromosome, okay, and with 40 picogram DNA. How much DNA it has? DNA is, that is, 40 picogram. What will be DNA here? Just double of this. That is in this. DNA will be equal to 80 picogram. Here also at G2, that is DNA will be equal to S. That is DNA will be 80 picogram. Is it clear? The right answer. Number of the chromosome will remain 40 and amount of DNA will become 80 picogram. Is it right? Okay. Come to some NCERT examples. Meiosis in a diploid organism results. Yes, it can make gamete. It reduces the number of chromosomes. It introduces variation because of crossing over. Right answer will be all of these. At which stage of meiosis does the genetic constitution of the gamete is finally decided? Very important, interesting question. Genetic, when the meiosis will divide into two, that is that here, because after this the gamete will be formed, they will remain same, their constitution will be almost similar, right? But here, that is genetic constitution of the gamete will be decided after anaphase first. Meiosis occurs in organism during sexual reproduction. Sexual reproduction, sexual reproduction is absent in case of, that is prokaryote. That's why prokaryotes, they do not have, that is meiosis, or we can say sexual reproduction. It is true sexual reproduction is absent in prokaryotes. They do not have meiosis. Mitosis is characterized by what? Reduction division? No. Equal division? Might correct both A and B. Is it? And this is pairing of homologous chromosome A and B. A and B, it cannot be there. Pairing of chromosome cannot be there. A means reduction division, no. Pairing of no, that is, it is only equal division. That is bivalent of meiosis consists of, 4 chromatids and 2 centromere bivalent. This is 1, this is 2. That is how many chromatids? 4 chromatids, and it has only 2 centromere, at which cells are not dividing. G0 phase. Wrong statement. 2 cycle of DNA. No, that is only 1 cycle of DNA replication. Select the correct statement about G1 phase. Metabolically inactive. No. It's not phase of no. Since DNA, this is the, that is correct statement about the G1 is DNA in the cell does not replicate, right? And see now previous year's some examinations question. Complex, that is synapse chromosomes are held together, that is known as bivalent. Enzyme recombinase related to pectine. Somatic cell just completed S phase of the cell cycle as compared to the gamete of the same. Right answer. Twice the number of chromosome and 4 times the amount of DNA. In meiosis, crossing over is initiated by, in which stage crossing over will be there? Recombination, that be in pectine stage. And see the last one, this anaphase promoting complex. Yes, a protein degradation machinery, that is APC is defective, not formed, then what will happen? Because anaphase promoting spindle fibers will not be formed, that is chromosome will not segregate. During which separation of the paired chromosome, that will be in diplotin, because synaptial complex will dissolve. Interphase includes, is this one, what is there, dividing cell, yes, enters vegetative inactive phase, this is called quiescent stage, and the process at this end will be, that is amophase. Meiosis, division of the secondary oocyte is completed, that at the time of, important this you have to study in, that is sexual reproduction also. Meiosis second of the oocyte, primary oocyte, secondary oocyte, you know the terms, that will be completed when the sperm will enter into the ovum, that time it will be completed. So this is, that is last part, part 5 of chapter 10. Thank you.